Hello, everyone. It's Steve Melito from Go Natural Education here. Today, we're talking to Dave Alburty and Dave Goad from InnovaPrep, a Missouri-based company that makes microbiological sample preparation tools. Dave and Dave, thanks for being here.
Thank you. Thanks for having us.
Great. So we're going to talk today about how and what InnovaPrep does and its relationship to farming, but I'd like to start with a statistic and some sound effects, if I may. And I'm going to go in reverse order because I'm not sure if you can hear birds chirping, but I'm working out of my home office here on the farm, and I've got some chicks in my office in a playpen. So hopefully they won't be too distracting, but I think it's sort of appropriate because the statistic I want to mention really caught my attention. It was in the news this morning, and it was about the bird flu, a really highly pathogenic variety that was detected at a large egg farm in Iowa. And there are 4 million chickens that are going to be destroyed as part of the effort to contain this disease. So animal health is clearly a very important thing for not just the livestock operator, but the family farmer as well. So I just want to set the stage there. And then to be clear with folks that InnovaPrep doesn't make detectors. What you make is you're on the front end of sampling and sampling preparation. Is that right?
Yeah, that's true. And we help avoid the garbage in, garbage out syndrome. If you don't have good sampling and sample prep, you don't get a good analytical result. And so you could easily miss something that's there and that could be a problem in your flock or your herd.
For sure. So let's talk a little bit about some of the larger livestock operations again, because they're in the news more often. When a large livestock operator suspects the presence of diseases, what do they do and where do your products come in?
Well, Dave's large scale, so I'll let him handle this one.
All right.
Large scale operators like feedlots and so forth, they typically have staff that veterinarians and so forth on call or on staff, and they'll take the samples and those will be processed. Usually, those are sent out often to animal disease diagnostic labs, and so that's where the actual detection process occurs. Some facilities have accredited laboratories where they can do actual sample testing. Where we fit in that is, as Dave mentioned, the other Dave. We've got too many Daves here. That we are involved with the concentration process. So we are really focused on being able to detect very small levels of particular pathogens. The Some facilities have. We'll go on to more details as we move forward, but
Sure.
some facilities are adopting this now. So.
Good. And if you're a small scale farmer like me, or even a rancher, is your technology simple enough for them to use, or do you need to really be a big operation with skilled technicians and folks like that?
Well, no, not necessarily. It can go all the way down to as small a scale as using our air samplers to collect samples and sending those in a mailer to a laboratory, kind of like you would with a mold sample at your house or a radon sample or something like that, where you would use a sampler for a limited amount of time when there's a concern and simply mail those into a partner laboratory where that then starts the sample tracking and reporting, and you end up getting your results back later. Not very much later. Everything's overnight email world nowadays.
Pretty much. So, Dave, you mentioned airborne pathogens, and so let's talk about a technology that you have called AirPrep. Did I get that right? Is that what it's called? Yes, it is. AirPrep for sampling airborne pathogens. And how does it work? And what are some of the types of pathogens that you would try to pick up for, say, chickens or cattle or hogs?
Well, sure, for any of those, they can be airborne transmission, the same as COVID is for humans. those can be breathed out, and you mentioned highly pathogenic avian influenza. That's a good example. In barns, it can be transmitted that way. And so their prep technology can be deployed to use our small samplers to collect the samples down at the ventilation end and send those in for analysis. And you can monitor constantly for anything to show up, whether it's that or any of the other viruses or bacteria that are of concern. Similarly, with most of the porcine operations, pig operations are very often operated out of barns, or the most critical portions of those operations are in barns, farrowing barns, and the hog barns. And so that kind of monitoring can use the air prep technology to detect something early and deal with it while maybe it's in one barn or at one farm, and start to take approaches to limit the spread of any kind of a pathogen or a disease.
Got it. Can you tell me more about the device, what it looks like, where you put it in the barn? Do you mount it on the wall? Do you put it on a table? How's it all come together?
Well, the AirPrep Cub is the simplest one to use, and it's the least expensive. It's about the size of a coffee can, maybe a little smaller, and you just unscrew the top and install the filter, which is about the size of a hockey puck. And you screw the top back on and turn it on it's got a timer, and you let it run. So it's got a little hanger. You can hang it up like a Christmas ornament down by the fan end of the house, or it's got legs. You could set it on a central location platform or floor even, or a table, and let it operate. When the time's up, you just collect the sample. Like you said, you put it in the mailer, and you can either hand it off to an on-site laboratory if you're a big operation, or you can mail it into the partner lab if you're a smaller scale. It's all dry. There's no liquid to worry about. There's no leaking or anything like that. That filter, is, I mentioned the hockey puck, is just it's a electrostatic collector, kind of like the ultra allergen filter that might be in your furnace. And because of that, it'll collect the particles all the way from the very smallest viruses all the way up to the exhaled breath particles containing bacteria, viruses, and other things that might be concerning.
Can you use this outdoors as well in, say, a pasture, or is this for indoor use only?
I know Dave had some use cases that we talked about that were outdoors and indoors.
And we've had some collaboration with researchers who looked at things like SARS-CoV-2 airborne in different areas, capturing that and being able to analyze those samples. Some people are working with environmental pathogens, viruses that come from really different kinds of sources. For example, there's one that involves bats, among other things, other types of animals. So there's a lot of different diversity in how this can be used. With respect to agriculture and sampling animals and animal facilities, the system is very, very easy to use. I actually show it to people and give them a very brief tutorial, and then they're able to just take off and run with it. So the ability to use that in lots of different animal scenarios is really good. I mean, it's really powerful. So it could allow you to do things like, of course, you think about bird flu, the monitoring and so forth, but there are other pathogens out there that I think have the potential to be used for this, like respiratory viruses in cattle, shipping diseases, BVDV, all kinds of things could be monitored with a system like this.
Good. Do you think
Yeah,
it could be used in, say, a feedlot, someplace where the animals are close together? Would that be a potential use case?
Yeah, I think it could be, as long as you have proper separation between animal lots and feedlots. They pen animals and they're put into basically grouped. And that's one of the things that the system has a lot of potential for, is that you can take samples from these different groups. If you have a sampler set up in a particular pen or in multiple pens, you can actually take that, and the product that comes out of that sampler could be pooled and then could be used for a larger area screening, and then go back and look at specific pins later. So it has a lot of power to be able to do not only at the pin level, but even larger than that. So
Good. And Dave Alburty, I think I cut you off there. My
Oh,
apologies.
I was just going to say, yeah, it can be used outside as well. And I think it was implicit in Dave's statement about feedlots. It's pretty hardened as far as rain goes or anything like that. I mean, there's a certain level at which you'll start to maybe ingest some water into the air sampler, but you could basically about dunk that thing in a bucket of bleach to sanitize it and keep your biosecurity rolling. So you can use it outside, you can use it indoors. It's been used in studies of, uh, like we mentioned, outdoor animal housing applications, feedlots, also been used in wild things like sampling around algae growth ponds to, determine whether there's GMO escape. Monitoring for GMO escape is maybe not something that's necessity for large animal growth, but it could be something agriculturally related, just pollen escape and transfer beyond what you were looking for on your farm.
Yeah, that's an interesting application. Can we talk more about that? Do you have a use case with that?
Oh, the one that I mentioned in particular, they took 4 samplers and they put them around an algae growth pond that was GMO algae, and they wanted to make sure that that wasn't escaping the location. And that was actually a state regulatory requirement that they had to do this monitoring.
Okay.
And so that one is a specific GMO air application that I know. I know of some animal applications. USDA put 6 of these samplers, the little air prep cub samplers. They put them around animal composting piles. And there have been some EPA studies too around animal composting operations. One of them was in a case of potential FMDV outbreak, and what would you do with, say, 1,000 cold cattle? And dig a long trench, put them in there, cover them up with compost, and try to compost them in place. And they wanted to understand if there was escape of the pathogen from that. So they simulated that. They didn't use actual FMDV, but they simulated an event. They were able to detect the cattle. Actually, it wasn't cattle. It was pigs. They detected the pig DNA kilometers away, even, but none of the virus surrogate even close by. So apparently, the composting did work. USDA did this with poultry at chicken farms in Georgia, and so they put 6 of these samplers around the composting chickens to determine whether pathogens were escaping the pile in an airborne fashion and exposing other chickens or the population, and determined that wasn't happening. But so I guess that's success. You want the process to be working, but what if it's not working? You could know if there's a problem.
For sure. Now you've also got a technology. We've been talking about the airborne aspect of it. There's a fluid sample preparation technology as well. Could we talk about that too?
yeah, that's the fluid prep system is focused on our concentrating pipette, as well as a larger liquid volume concentrator. We call it LVC, large volume concentrator. I'll let Dave talk a bit about that. The concentrating pipette is able to take samples, liquid samples, up to depending on the quality of the sample, it can be up to about 3 liters, and then concentrate that down into a volume that is less than a milliliter. So usually half of a milliliter, 0.2 milliliters. And so that you have a very high volumetric concentration efficiency, and that allows you to detect pathogens that are in very low numbers. You know, I just had a project with marine pathogens, and being able to detect 10 pathogens and 10 to 100 pathogens in a liter of seawater is pretty significant. So it has a lot of utility there, and we can talk about how that functions. I'll let Dave talk a bit about how the system operates, but it has been used in just as a lead-in, I guess, a lot of wastewater analysis. So human or wastewater treatment plants and wastewater associated with SARS-CoV-2. Wastewater testing became really a really powerful tool for community surveillance, community monitoring during the SARS-CoV-2 pandemic. And that's because the signature that you see in wastewater tended to precede the kind of events that you would see at a clinical level. In other words, people coming in sick. It would precede it by anywhere from 4 to 10 days. And so it was a great tool. Those same strategies can also be employed with animal health. For example, looking at wastewater from runoff or whatever from animal facilities, there's a potential to detect pathogens that way as well. And I'll turn over to Dave to explain how the system works. He can do that much better than I can.
Sure. Well, sure. Yeah, absolutely. The concentrating pipette or the LVC both work the same way. The liquid sample is just pulled into the lumen of hollow fibers that are in a tube. So it's like leaky soda straws inside a tube. You pull the sample into the leaky soda straws, and they capture the particles that you're interested in, the viruses or bacteria. Once that's processed and those are captured, then it's eluted back out of the bores of those hollow fibers, the lumen of those hollow fibers, using a wet foam, which is a carbonated surfactant buffer. Think soapy Pepsi, salty soapy Pepsi. And as it pushes the particles out of the bores of those fibers, it collapses again. It wants to go flat right away. And so there you have this tiny sample. You can suck a whole bunch of sample through it and then basically elute that or spit that out in a very small amount of fluid for analysis. And so it makes it a powerful tool for quickly concentrating samples. Either Dave mentioned marine applications, and some of your listeners might be into aquaculture. It's a big deal in farming. So whether for land-based or sea-based or oyster farming, protecting your animals in the aquatic environment is, would be concentrating the air the same way we might sample air for cattle or pigs. That's what they breathe. Another thing I might mention about pooling that Dave described is, these air samples can be pooled, and you could take several air samples that are flushed from that dry filter with a liquid for analysis and then concentrate that. So you can really magnify your sensitivity for sampling around some large area using this instrument. So it would be something that would probably be in a laboratory at a large scale livestock production lab or a veterinary lab or a clinical lab that would then be applied to these samples that would be sent in, either from an aquaculture basis or from air sampling basis or from even sampling things like irrigation water, because everything's connected to everything. In a One Health environment that we're talking about here, people are connected to animals are connected to people through the air, through the water, through the surfaces, through the land, through the recycle loop, the poop loop, Dave calls it. And so just because everything's connected, we look all across the spectrum of that front end to try to help deliver the results at the end of the day for the proper kind of analysis.
One thing that I mentioned and Dave mentioned, LVC, the large volume concentrator. That one's interesting because it can handle volumes up to 100 liters. You can take, just as he mentioned with the air concentrators, you can take what comes out of that and concentrate even further with a concentrating pipette. You can do the same thing with a large volume concentrator. So you can do sequential concentrations and essentially get down to a less than 1 milliliter that has everything that came out of 100 liters of original sample, original water, or for example, if you're looking at irrigation waters or something like that. So it's a really powerful concentration sample prep tool.
Sure is. It sure. And I wanted to ask you about soil because, as you said, everything's sort of connected together in that poop loop. Can your technology at all be used to look for pathogens or microorganisms in the soil samples? Or
Yeah,
help
absolutely.
prepare? Okay, yeah.
Yeah, well, yeah, that's pertinent too, right? Because dust, for example, in a livestock facility, is one of the ways that these things are spread. The animals will, as part of the poop loop, they'll do their thing, and then they'll do their thing, and then that'll get stirred up into the air, and the animals breathe it. And but just as far as the soil itself goes, you can take that and put that into a liquid, mix that up, and let the non-target silt particles settle out, so forth, or do a pre-filtration step to clarify that so that you can concentrate the bacteria and viruses from that. You kind of basically make the soil into like a wastewater type of a liquid, and then you can run it on the concentrating pipette, concentrate that down for analysis. You're kind of extracting the biological materials from the soil first and then concentrating them.
Yeah, so we've talked a lot about animal health. Can we talk about plant health as well? And do the technologies you have, can they be used in, say, a greenhouse or even outdoors in a field somewhere?
Yeah, I took one of the cub samplers to a medical marijuana growing facility. It's the biggest single, single crop greenhouse I've ever been in. Every other greenhouse I've been in is growing lots and lots of things. And you can sample the air in there and look for pest, organisms. And in the marijuana facility, they were looking for the hop latent viroid, and they were looking for pollen because they were growing all female plants. They wanted to find out if a hermaphrodite plant had formed somewhere and was pollinating these females because that would decrease the value of their crop. And so it was really a nice application to use that air sampler in, and you could collect at all of the different portions of the facility, pool those, concentrate those, look for the target hop latent viroid and the pollen. I did find one pollen grain on that experiment, so they did have a hermaphrodite somewhere in their operation, and they probably had a hunter-killer mentality about that to find that thing. But this in the greenhouse operation too, they suspect that this hop latent viroid is actually spread by their irrigation water supply because they recirculate a lot of that water. They filter it, they do things to it, they use it for the fertilization and the hydroponics. But that's one of the areas of suspicion. And so between doing the air sampling and the water sampling, you can gain control of your environment in those large greenhouses, whatever kind they are.
Got it. And Dave Goad, anything that you would add when it comes to plant health and InnovaPrep's technology?
Um, the irrigation water that he talked about, that's significant. The and then the looping back to soil analysis, the soil analysis is important for plants as well. So especially from the standpoint that a lot of the pathogens in plants are virus related, and so that it's difficult sometimes to collect those, to find those. And our technology is very effective for being able to concentrate virus and present that to a downstream analytical process, whether that be polymerase chain reaction, PCR, quantitative PCR, even sequencing is possible with the samples that come from our system with the proper downstream processing. So,
Yeah, USDA did a study in 2018 and showed the efficacy of that exact approach.
Excellent. Any other case studies or use cases you'd like to mention that I haven't asked you about or given you an opportunity to discuss?
I would say one thing a lot of folks use our equipment for is what happens after the farm. It may be farm to fork to pharmacy. Who knows, right? So um, looking at the food itself that's produced. And so there've been a lot of success in looking at food in food processing plants. One of them recently is looking at norovirus from berry production in where they freeze the produce. They take fresh produce, they want to sample that. It could be frozen and they want to sample that. It could be leafy greens or another favorite culprit for E. coli varieties. The instrument's really good for taking those sorts of wash waters or rinse waters, sampling from the food and giving a ability to do a quick and sensitive analysis so that you can get a result same shift and release the food on down the line so that you're not waiting and holding it, waiting on your results or trying to recall trucks or there are many strategies for trying to increase the shelf life, and one of them is to try to get your analytical result as fast as you can. And so that's where we can come in to help. And if you think about that even more and you're able to reduce refrigeration, now you're talking about having a positive impact on climate change, certainly carbon footprint and things like that. So it may have, beneficial impacts in ways you can't foresee when you first start looking at the use of those products.
Right. And when you think about how your technology compares to what's available in the marketplace, is the speed and the ease of use what makes you different? What are the biggest pluses that you bring to the table?
I would agree. Our customers have the need for speed. That's the main thing. And our products are used by the military too, and so they won't use something unless it's easy to use. Anything that's too complicated is just a non-starter for most military applications, you know, in this field. And so the speed, the ease of use, and then because of those two things, it can save you money in a lot of applications. An overlooked thing in a lot of laboratories is how much technician time and frustration really costs them. If you can make their job easier, that makes the manager look good. And if you can make the manager's job look easier, that they feel good about that. And if you can save them money, that matters to the CFO. And so you can just, kind of benefits all up the line because of those three things.
Yeah, for the applications like your wastewater, stuff that would be farm related, that looping back to the human side, the system that we have with the concentrating pipette and the methods that have evolved have taken a process that can, in some cases, it's a 2-day process, the older methods, but in general, processes that take around 2 hours or so, it condenses that into a time span of 10 minutes. Our system can do things much faster in terms of the concentration, the viral concentration. Like I said, for the SARS-CoV-2 during the pandemic, with wastewater, that was it increased workflow tremendously for a number of public health labs. So it became a really popular method, and the same holds true for other methods that you would compare it to. So the speed factor is important. The other thing that's important also, though, I think, is the ease of use. So the system, all our systems are designed to be simple to use for operators, and the need for a dedicated technician or somebody with a high level of technical expertise, it's not there. You don't need it. So you can come in and take your sample, concentrate it, and as Dave mentioned with the InnovaPrep system, you can load the sample up, start it, run it, pull the collection filter unit out, drop it in an envelope, and send it along the way to a test facility. And it's really it's all those types of things are pretty simple to do.
Excellent. And sort of last question here, if there's a smaller farm out there that is interested in your technology, wants to learn more about it, what should they do? Go to your website, reach out to you? How do you like to interact with folks?
Yeah, the easiest way is to reach out to the website and go to customer service and talk to somebody. And you can actually talk to a real person, which would probably be a relief to them.
For sure. For sure. All right, Dave Alburty and Dave Goad from InnovaPrep, thanks so much for being part of the Go Natural Education podcast.
Thank you.